Hi, in today's video we will be talking about carcinogenesis. In carcinogenesis we will be touching upon the concepts of chemical carcinogenesis, physical carcinogenesis and biological carcinogenesis. So chemical carcinogenesis, the first ever evidence of cause of neoplasia was from the observation of Sir Percival Pott. He observed that there was a very high incidence of cancer in the chimney sweets. Which cancer? The scrotal cancer. So that is when he realized that there can be an association between the soot and the coal tar leading to cancer and possibly other occupational cancers as well. Alright. So the first successful experimental induction of cancer was produced by two Japanese workers, Yamagiwa and Ichikawa, in 1940 in the rabbit skin by repeatedly painting with coal tar. This is Mr. Percival Pott. He was an English surgeon. So what are the steps in chemical carcinogenesis? We will see, and we will see also the carcinogenic chemicals in humans. Now, stages in the chemical carcinogenesis. Induction of the cancer can occur by chemical carcinogens after delay weeks to months in case of experimental animals and often several years in man. That's why we say when you smoke, immediately you don't get cancer, but after a period of about 10 years, the incidence of cancer, bronchogenic cancer, oropharyngeal cancers increases. Now, what are the factors that influence the induction of cancer? It is the dose and mode of administration of the carcinogenic chemical, individual susceptibility and predisposing factors. The basic mechanism of chemical carcinogenesis is by induction of mutation in the proto-oncogenes and anti-oncogenes, and the phenomenon of cellular transformation by chemical carcinogens progresses through three famous steps: initiation, promotion, progression. We will see what are these three steps. In initiation, there are different factors involved like metabolic activation, reactive electrophiles, target molecules, and then there is an initiated cell. So we'll see that in detail. The next step is promotion followed by progression. So what happens in initiation is there are direct acting carcinogens, there are indirect acting carcinogens which are called procarcinogens. The direct acting do not require any further activation, whereas the indirect ones require metabolic activation to target the target cells. In the target cells, reactive electrophiles are formed which identify the target molecules in the DNA and cause permanent DNA damage. This all is part of initiation. Whereas in the step of promotion, we have clonal proliferation of altered cells leading to cancer phenotype leading to a malignant tumor. So initiation is the first stage in carcinogenesis induced by an initiator chemical carcinogen. The changes can be produced by a single dose of initiating agent for a short time. Though larger dose of long duration is more effective, the changes are sudden, irreversible and permanent. The chemical carcinogens acting as initiators of carcinogens can be divided into two categories: direct acting carcinogens and indirect acting carcinogens or procarcinogens. So, which are the direct acting carcinogens? In them we have the alkylating agents and the acylating agents. These cause cellular transformation without any prior metabolic activation. The indirect acting carcinogens or procarcinogens require metabolic conversion, and among the prototypes we have polycyclic aromatic hydrocarbons, aromatic amines and azotides, naturally occurring products and miscellaneous. So what is metabolic activation? This is in the initiation of carcinogenesis. So majority of the chemical carcinogens are indirectly acting or procarcinogens requiring a metabolic activation. They get activated in the liver by the monooxygenases of cytochrome P450 in endoplasmic reticulum. The metabolic activation has two requirements to determine the carcinogenic potential of a chemical. First, the balance between the activation and the inactivation of the carcinogen chemicals. Second, the genes that code for cytochrome P450 dependent enzymes involved in metabolic activation. Next, coming to reactive electrophiles, these are the direct acting carcinogens are intrinsically electrophilic. Indirect acting substances become electron deficient after metabolic activation. That is, they become electrophiles. Phile means love the electrons. Because they are deficient in electrons, they love the electrons. They bind to electron-rich proteins of the cell, the DNA and the RNA and other proteins. What are the target molecules? The primary target of electrophile is the DNA, which produces the mutagenesis. This leads to the changes in the DNA to the initiated cell. Precancerous conditions like xeroderma pigmentosum are prone to skin cancer. That is an example of an initiated stage. Most frequently affected promoter oncogene is RAS gene mutation. Remember, the anti-oncogenes is P53 mutation. Once there is an initiated cell, you have unrepaired damaged DNA which becomes permanent and fixed only if altered. Cells undergo at least one cycle of proliferation, results in transferring of change to the next progeny of cells, so that the DNA damage becomes permanent and irreversible. That is the characteristic of an initiated cell. Remember, in an initiated cell, the DNA damage is permanent and irreversible, vulnerable to the action of promotion of carcinogenesis. What are the stimulus for proliferation? It will be regeneration of the surviving cells, dietary factors, hormone-induced hyperplasia like endometrial hyperplasia leading to endometrial cancer, estrogen on the breast cancer, hepatocellular cancer arising from viral hepatitis. All these are examples of initiated cells. Coming to the second stage, which is the promotion of carcinogenesis, is the next sequential step in chemical carcinogenesis. Example is formol esters, phenols, hormones, artificial sweeteners, phenobarbital. They do not differ much from the initiator. Sorry, I beg your pardon, they differ from the initiator, as in they do not produce the sudden change and they require application from initial exposure for sufficient time and sufficient dose. So, how are they different? The changes induced may be reversible. Remember, the initiated cell is irreversible. Promoter changes are reversible. Do not damage the DNA per se and thus not mutagenic. Instead, enhance the effect of direct acting carcinogens or procarcinogens. Act by further clonal proliferation and expansion of the initiated cell. Have reduced requirement of growth factor, more so after a RAS gene mutation. So this is a very famous example taken from Robbins. Robbins is the Bible for pathology. So what does it say? We will see it again, right? So only initiator, no tumor. Initiator followed by promoter, there is a tumor. Initiator with the gap followed by a promoter, there is a tumor. Lot of promoters followed by initiator, no tumor. Only promoter, no tumor. Initiator followed by a promoter at a good interval, no tumor. That's why remember I told you promoter has to be applied at a very repetitive rate to have an effect. So the next step is the progression of carcinogenesis. It's a stage when the mutated proliferated cell shows phenotypic features of malignancy. Features pertain to morphology, biochemical composition and morphological features of malignancy. The phenotypic features appear only when the initiated cell starts to proliferate rapidly and acquires more mutations. New progeny of cells that develop after such repetitive proliferation inherit genetic and biochemical characters of malignancy. So, based on the mode of action, they are divided into two groups, the initiators and the promoter carcinogenesis. We have the direct acting, which are the alkylating and acylating agents, and the indirect acting, which are called the procarcinogens. We have the polycyclic aromatic hydrocarbons, aromatic amines, and azodyes, naturally occurring products, miscellaneous. In the promoter carcinogens, we have phenol, formol esters, hormones, and miscellaneous. In the initiator carcinogenesis, we have direct acting, do not require metabolic activation like alkylating agents. Weakly carcinogenic, implicated in the etiology of lymphoma and leukemias, anti-cancer drugs like cyclophosphamide, chlorambucil, beta-propylactone, epoxide, acylating agents like imidazole, dimethyl carbamyl chloride. The initiator carcinogens are also called the procarcinogens. They comprise of a large group of procarcinogens implicated in a number of human neoplasms, cause different effects by various modes of administration. Topical application can lead to skin cancer, subcutaneous injection can lead to sarcomas, inhalation can lead to lung cancer. Indirect acting procarcinogens, polycyclic aromatic hydrocarbons, main source is combustion, chewing tobacco, smoke, fossil fuel, soot, tar, mineral oil, smoked animal foods and pollutants. Important compounds included are anthracenes, benzopyrenes, methyl, chloranthrene. Indirect acting carcinogens or the procarcinogens, they are polycyclic aromatic hydrocarbons with evidence to support etiological role of these substances. Smoking and lung cancer. Now 20 times higher incidence of lung cancer is recorded in smoking of up to 2 packs per day for 20 years. Skin cancer, direct contact with skin associated with high incidence of skin cancer. Remember the Kangri cancer? Kangri is a bowl of charcoal which is tied around the abdomen, very common in the Kashmir area to prevent from the frightful and the cold environment. But that is a cancer and that leads to cancer of the abdominal area, the skin of the abdominal area. So tobacco and metal chewing very popular in the subcontinent, Indian subcontinent, can lead to oral cavity cancer. Indirect acting carcinogens are the procarcinogens like aromatic amines, azodyes. Beta-naphthylamine can cause bladder cancer with aniline dyes and rubber industry. Azodyes like the coloring foods, butter and margarine to give them a yellow color, scarlet, red for the coloring cherries can all lead to hepatocellular carcinoma. So be careful of the colored food. You may enjoy the tandoori chicken, which is red in color, but that is all not very good, right? Naturally occurring products are the plant and the microbial sources like the aflatoxin, actinomycin D, mitomycin C, saffron, betel nuts. Aflatoxin in particular can lead to hepatocellular cancer, and that is because of the fungus Aspergillus flavus that grows in the stored grains and plants. Coming to the indirect acting carcinogens. So coming back to the indirect acting carcinogens, which are the procarcinogens. So then we also have the miscellaneous category within them, the nitrosamines, which cause the gastric cancer, and the vinyl chloride from causing the hemangiosarcoma of liver, asbestos leading to mesotheliomas, arsenical compounds causing basal cell carcinoma, metals like the nickel and the lead causing the lung cancer. So based on the mode of action, again we divide them into two groups, the initiators and the promoters. So direct acting, again to recap, the direct acting are the alkylating acylating agents, whereas the indirect acting are the procarcinogens like the polycyclic aromatic hydrocarbons, aromatic amines, naturally occurring products, miscellaneous. Promoter carcinogens are the formol esters, hormones and miscellaneous. Next, so we have discussed this. Promoter carcinogens are chemical substances which lack the intrinsic carcinogenic potential, but the application subsequent to initiator exposure helps the initiated cell to proliferate. Example are the formol esters, hormones and miscellaneous. Coming to physical carcinogens, so we know that they are the radiation carcinogenesis caused by ultraviolet light, ionizing radiation, and the non-radiation carcinogenesis injury. So the radiation carcinogenesis are the induced mutation followed by a long period of latency. After an initial exposure, often for 10 to 20 years, they act to enhance the effect of another carcinogen called the co-carcinogen. The UV light and the ionizing radiations differ in their mode of action. So ultraviolet light is the main source of sunlight, penetrates the sun only for a few millimeters. Effect is limited to the epidermis, and the efficiency of UV light as a carcinogen depends upon the extent of the light absorbing protective melanin pigmentation of the skin. Excessive exposure of the UV light leads to skin cancer like squamous cell carcinoma, basal cell carcinoma, and melanomas. UV light mechanism is the induction of mutation. Others include inhibition of cell division, inactivation of enzymes and cell death. Important biochemical effects are the formation of pyrimidine dimers in DNA. Normal individual DNA damage is repaired. Zero double pigmentism, I told you, is a condition when there is a defective DNA repair mechanism that is associated with the high incidence of skin cancer. Then coming to the UV mechanisms, again I told you, the other conditions are like ataxia telangiectasia, predisposing to leukemias, Bloom syndrome, okay, leads to all type of cancers, Fanconi's anemia, increased risk to develop cancer. Coming back to ionizing radiation, the X-ray, alpha and beta gamma rays, the radioactive isotopes, protons, neutrons, they all induce leukemia except CLL and papillary carcinoma of thyroid. Risk is increased by higher dose with high linear energy transfer neutrons and alpha rays. So ionizing radiation evidence in support of carcinogenic role of ionizing radiation, high incidence of radiation dermatitis, subsequent malignant tumors of the skin, high incidence of osteosarcoma. So especially in the young American work girls. So what they used to do, the girls who were working the American the dye industry and all that, they used to just have the bad habit of licking it. When they were licking the paint, as a result of it, they ingested the cancers, carcinogens, and that led to the cancer. So especially those engaged in painting the dyes with luminous radium unknowingly ingested radium while using lips to point their brushes. Then other things are the miners in the radioactive elements have the higher risk of cancer. The Japanese atom bomb survivors of Hiroshima and Nagasaki, quite famous, they all developed cancers, notably AML, CML, solid tumors of breast, colon, thyroid and lung. Accidental leakage happened at the nuclear power plant in Chernobyl that caused a long-term hazardous effect. Alright, and therapeutic X-ray radiation resulted in increased frequency of cancer. And what about the other things? So RNA damages the DNA by one of the two mechanisms. It can directly alter the cellular DNA or be dislodged from the water, ions of the water, and other molecules that results in highly reactive free radicals that bring about the damage. And then there is damage to the DNA resulting in mutagenesis, chromosomal breakage, translocation, point mutations, and it depends on the type of rate, dose, and dose rate frequency. Other factors to look into are the age, individual susceptibility, immune competence, hormonal influence, and types of cell irradiated. So following is the exhaustive list of cancers that can result from the radiations and the non-neoplastic effects like leukemia, carcinoma of thyroid, carcinoma of breast, carcinoma of skin, carcinoma of lung, cataracts, pulmonary fibrosis, myocardial fibrosis, constrictive pericarditis, strictures, nephritis, congenital malformation, sterility, and radiodermatitis. Coming to the non-radiation carcinogenesis, okay, so sometimes physical carcinogenesis we also call it, like stones in the gallbladder can cause the gallbladder cancer, stones in the urinary tract, healed scars following burns or trauma can all lead to cancer. Coming to biological carcinogenesis, important topic again. So here we have the viral oncogenesis and the DNA viral oncogenesis and the RNA viral oncogenesis. So it's a question usually asked in the exam, name the viruses causing DNA, cancer. So you can classify them as the papilla or the DNA oncogenic viruses. In this you have the papillomaviruses, the papilloma, polyoma, SV40. Then the next category is the herpes virus of Epstein-Barr virus, human herpes virus 8. The third category is adenovirus, followed by pox virus and hepatDNA virus. So the specific RNA oncogenic viruses are the acute transforming viruses, the slow transforming viruses, and the human T-cell lymphotropic viruses. And so viruses and human cancer are summary. So we can have some benign conditions, some malignant conditions. So parasites, schistosoma hematobium in the bladder can cause squamous cell carcinoma. Trilogus sinensis in the liver can cause cholangiocarcinoma. Aspergillus flavus in the liver can cause hepatocellular carcinoma. Helicobacter pylori can cause gastric lymphoma, gastric carcinoma. Cervical cancer is very important and is now a preventable form because it can be early detected. There is a vaccine available. You can do a pap smear and detect it, right? So that's caused by human papillomavirus. So let's talk about the papillomaviruses. There is a low risk papillomavirus, HPV, type 6 and 11, which causes the genital warts of the condyloma acuminata. And we also have a high risk HPV, 16, 18, 31, 33, and 45. These cause cervical cancer in 75 to 100%. The high risk HPV can also spread to the anus, causing squamous cell carcinoma in the perianal region, vagina, vulva, penis, and the oral cavity. So persistent infection with high risk HPV drives the molecular hallmarks of cancer, directly affects the cellular growth by following mechanism. HPV integrates into the host cell DNA, resulting in overexpression of E6 and E7 from high risk HPV types. E6 and E7 have high affinity for target host cell than the low risk forms. E6 and E7 cause loss of P53, retinoblastoma, and these two cell proteins have the tumor suppressor properties. So they suppress the tumor suppressor proteins, right? And these viral proteins activate cyclin A and E, inactivate CDKIs, thus permitting the further cellular proliferation. Viral proteins mediate and degrade PAX, a pro-apoptotic gene, and thus inhibiting apoptosis. Viral protein activates telomerase, immortalizing the transformed host target cell. Right? Epstein-Barr virus infects the human B lymphocyte and epithelial cells. Its long-term infection stimulates them to proliferate, develop malignancies, cause infectious mononucleosis, self-limiting disease in the humans. EBV is implicated in Burkitt's lymphoma, angioplastic nasopharyngeal carcinoma, post-transplant lymphoproliferative disease, primary CNS lymphoma in AIDS, and Hodgkin's lymphoma. Coming to Burkitt's lymphoma, initially noticed in African children by Burkitt in 1958, known to occur in two forms, African endemic and sporadic form. Burkitt's lymphoma is one lymphoma which can be easily treatable, though it's very aggressive. If you diagnose it quick, it is amenable to chemotherapy. So strong evidence linking Burkitt's lymphoma with EBV from the following features. 90% of Burkitt's lymphoma are EBV positive, in which the tumor cells carry the viral DNA. 100% cases show elevated levels of antibody titers to various EBV antigens. EBV has strong tropism for B lymphocytes. EBV infected B cells grow in cultures and are immortalized. EBV infection is worldwide in adults and causes self-limiting infectious mononucleosis. But the fraction of EBV infecting circulating B cells in such individuals are very small, or extremely small. Linkage between the Burkitt's lymphoma and EBV infection is very high in African endemic form than in the sporadic form. In sporadic cases, immunosuppression plays a significant role. Evidence of fever. It is observed that malaria, which confers immunosuppressive effect on the host, is prevalent in the endemic proportions in regions where endemic form of Burkitt lymphoma is frequent. This supports the linkage of EBV infection and immunosuppression in the etiology of Burkitt's lymphoma. EBV can also cause the anaplastic nasopharyngeal carcinoma, right? So the tumor is prevalent in Southeast Asia. 100% cases of nasopharyngeal carcinoma carry DNA of EBV in the nuclei of tumor cells. Individuals with this tumor have high titers of antibodies to various EBV antigens. Coming to human C T cell leukemia virus is a retrovirus causing T cell leukemia. It stacks proteins, inactivates the cell cycle inhibitors, and enhances cyclic T, thus dysregulating the cell cycle. So, thank you for your kind attention. So, this was a very quick wrap up of chemical carcinogenesis. So, we touched about chemical carcinogenesis, physical carcinogenesis, and biological carcinogenesis. In chemical carcinogenesis, we studied about the initiation, promotion, right? What are the initiators we studied? We studied about the promoters. In the physical carcinogenesis, we saw what are the radiations, the X-rays, the gamma rays, which can cause cancer. And then we studied about the biological carcinogenesis, the viruses, the DNA viruses and the RNA viruses. Don't forget to like and subscribe the channel. Thank you.